Okay, guys, let's talk about enzymes. Now, enzymes, don't forget, right? Let's go through all the stuff we know already. So, enzymes, these are going to be a type of protein, right? A special class, I guess, is what they are. Whoa, that spells special, right? So, these are a special class of proteins. They work whenever it's folded correctly. So they work whenever they get to that tertiary structure. And remember, when we talk about structures, this tertiary one, this is formed by the interactions of the R groups. Because, as just a tiny little, remember, primary structure is just, well, let's just do the polypeptide chain. So polypeptide chain. And then secondary structure is when either you get the alpha helices or you get the beta pleats. And these are held together by hydrogen bonds. And then we get our tertiary structure, which is the one I want right now. And tertiary structure is when I have those helices and those pleats, and these are going to be interactions of that R group. And whenever we get to those interactions of the R group, that's when you get that globular shape, the one that we did with the pipe cleaner and the beads, and it looks something like this. So even though when I draw an enzyme later here in the notes, it usually looks like Pac-Man, it's really more something along those lines. Okay, so let's go back to a few more definitions. And he's also known as a biological catalyst. Now remember, a catalyst from chemistry, this is something that speeds up a reaction. And this is a biological one. Remember, we always read things backwards. This is a biological one because bio means life. So this is a molecule, a protein, however you want to describe it, that speeds up reactions in living systems. And so how my enzyme speeds up reactions, well, we have a couple different ways. Really, the definition for an enzyme is it speeds up a reaction by lowering the activation energy. And when I talk about activation energy, this right here, this is just the energy needed for a reaction to take place. All right. And, we've drawn the hill with the balls before, so it's really the easiest one. So let's say this is hill A. So hill A. And then we have hill B, something like that. And our job, regardless of where we are, is to get the ball from the starting point to the end point like this. And so if I look at it, it's fairly simple, but which one is going to take less energy? Well, that's going to be A, because I only have to do that. B is going to take a lot more energy because I have to go all the way up the hill and then back down the hill. Can B still happen? Can this reaction right here still happen? Of course, but this is going to take more energy, right? And this is going to take more time. Versus this guy right here, A, this is going to take less energy and it's going to take less time, which is the very definition of an enzyme, because an enzyme speeds up a reaction less time by lowering the activation energy, which is the less energy. So even though when we talk about these reactions inside a living system, right, when we talk about a reaction, they can happen without an enzyme. But here's the deal, is that since this is in a living system, do we really have time to wait for it? Because, living systems, we usually have to have those reactions pretty quickly to keep things in order, so that way we can keep our living system going. Now the reactions we're looking at, remember, these are my hydrolysis reactions, and these are these are my dehydration reactions. And remember, during hydrolysis, I am breaking things down with water, and then during dehydrations, I am building things with water, okay? So whenever these enzymes do lower activation energy, it's usually because they do a couple points, right? So if we draw a really easy example, like let's say I want to do dehydration. And so here's my molecule, right? This guy right here. So this is my enzyme. And hopefully remember from whatever biology class we had last year, that right here, this is called my active site. And then whatever the reaction is, because let's just think about like a normal chemical reaction. So I have my reactants, and then I make a product, right? So these reactants, these actually have a special name now. Reactants are called substrates. Let's spell it out. Substrate. And then the products, these are still going to be a product. And so over here, this is my enzyme. So, and what am I doing? Dehydration, I'm building. So then this is my substrate, and this is my substrate. And the entire goal is to get these two to come together. So I want to build something. And so what happens is, here's my enzyme. I'll just make it with a big E. Here's my active site. And now these two substrates, they go in and they sit inside the active site. So this enzyme is literally orienting the substrate to be closer together, right? Like, could this substrate and this substrate at some point pass by close enough to each other that they want to interact? Of course they can. But again, we're not really willing to wait on chance. And so that's why we have enzymes. Enzymes are going to facilitate, they're going to make this favorable environment for the substrates to interact with each other a lot more quickly, right? So then this right here, this is called my enzyme substrate complex. And so now that reaction is going to take place. And so here's my enzyme. And now, because I was doing dehydration, now they're one thing together, and this is my product, right? So all of these chemical reactions that we've been talking about and that we're getting ready to talk about, the enzyme is just facilitating the reaction, right? Enzymes, let's write it down. Enzymes, oh, come on, I wanted that pretty color. There you go. Enzymes facilitate reactions, okay? They just make them happen. And of course, I can do the reverse of this. So let's do hydrolysis. So now I'm going to break something down. So here's my enzyme. Here's my active site. And then this right here, this is my substrate. This is the thing that I want to do the reaction to. So, enzyme, active site, and then the substrate comes and sits inside. So this is my enzyme substrate complex. And then, of course, because that enzyme, again, he's going to orient those substrates. He's going to make this favorable environment for the reaction to take place. Maybe this enzyme, maybe what he's doing, in his tertiary structure is taking this substrate, and maybe some of those amino acids, some of those R groups have different charges, and that's going to rip this substrate apart, right? I mean, again, it kind of depends on the reaction. So then here's my enzyme, here's my active site. And so now, because this was a hydrolysis reaction, I get two products because I broke something apart. So again, all the reactions that we've ever done, now we're just having a place for them to happen, right? It's kind of like a dating site almost. Like if you have this person who lives, let's say this person lives, I don't know, this person lives in LA, right? And then you have this person right here, this person lives in Texas. All right, is there a chance that these two could meet just in normal circumstances? Sure, but the odds are low. But now that you have these awesome dating sites, right? Dating sites, they can put their profiles on there, they can match, and then their chances of meeting, look at that, see, now they've met, those chances of meeting are much more likely because of the matching. And so now you get your product versus this over here. If we didn't have the dating site, is there a chance they can meet? Sure, but it would take a lot longer and the likelihood of it happening is really low. It's the same thing with chemical reactions. Like this substrate up here, like, is it possible that he might just meander into the right conditions and he would break apart? Sure, but since we're trying to keep a living system alive, we can't really wait on it. Now, a couple other things about enzymes. Not a couple, we have a few more things. One thing about enzymes is they always end in the suffix ase, just while I'm thinking about it. And usually they're named off after the thing that they're doing the reaction with. So like, let's say I have sucrose, right? So sucrose we know is a sugar because it ends in OSE. And then the enzyme that's going to break sucrose apart, this is called sucrase, right? If we wanted to do, I don't know, another sugar. So if we want to talk about lactose, again, another sugar because it ends OSE. And then the enzyme that breaks lactose apart, this is going to be lactase, right? Are they always so easy? No, because if I want to break a protein apart, breaking proteins apart, that's actually called pepsin. So do they always follow the ACE stuff? No, but they pretty much do. Now, if I'm looking at, let's talk about the lock and key model first. Okay, so lock and key model. And hopefully you guys remember this from Gen Bio last year, but really all the lock and key model is that these enzymes are specific to the substrate, right? And so if this were my enzyme, right, this is my enzyme, and I have these substrates to choose from, triangle, square, circle, well, it's just like the little kid game. Which one's going to fit into the active site, guys? It's the triangle because he fits, right? That's the lock and key model, because the whole idea behind this is that if I have specificity, then that means I get the reactions to happen faster, right? If I had an enzyme like this and all substrates were triangles that could fit into that active site, well, would the reactions happen? Sure, but what if I needed this specific triangle to do the reaction before this guy over here? There's nothing stopping that reaction from taking place or that reaction taking place before this guy. And if I'm waiting on that reaction to have some type of biological impact, again, to keep my living organism alive, then I might die because I keep waiting for the one reaction to take place. But since my enzymes have specific shapes, have specific active sites, that is fit for my substrate, then I get to do the reactions a lot more quickly because it's specific. Okay, and that's where the lock and key model comes in. If I have my front door key, so here's my front door key, and that's going to fit my front door, right? Like that's a check. But then if I take my front door key and I go to my neighbor's front door, Well, just because it's a key and it's a door, that doesn't mean it's going to fit, right? So again, it's very, very specific, but it's specific because of generally the active site, because the active site is going to be a certain shape. Again, remember, this is my tertiary shape, so it's going to form a very specific shape where the substrate can fit in, and then once it can fit in, then the reaction can take place. So now, when we talk about that enzyme, right, and we talk about the active site, there's another idea that we didn't talk about last year, at least I didn't. This is called induced fit. And really what it is, it's kind of precious that if I have my enzyme and I have my substrate, and then when that substrate fits into the enzyme, because of the interactions of the R groups on my substrate, this enzyme kind of gives him a little hug. Like, he goes and fits in there, but then it's like, nah, like I'm gonna grab onto you super tight. That's called the induced fit, because your enzymes will change shape just a little bit, not a whole lot, but just a little bit, so that way the substrate fits in there better. And if the substrate fits in there better, then your reaction can happen even more quickly, right? If we wanted to draw a graph of this, right, we kind of did it with the hills. This would be the rate of reaction, and this would be energy needed, right? So let's do the first one in blue. So here's my starting point, and then that's the energy, and then this is where the products are formed. So products. This guy right here, this would be with my enzyme, because the energy is going to be lower, even though if I start off at the same part, and then those even out, this is without my enzyme. So again, even though I reach the same end goal, I want to be able to do it with the least amount of energy versus this guy, because the more energy I spend, then, just the less efficient my organism's going to be. Now, things that can affect my reaction rate. Okay, so these are going to be things that affect reaction rate. Affect reaction rate. Well, remember, this is a protein, and so proteins are always like my puppy. What are things that can damage my puppy? All right, well, if I put the puppy in an oven, right, the puppy dies. If I put the puppy inside the freezer, the puppy dies. And so temperature has a huge effect, right? So if I raise or if I lower the temperature, that's going to have an effect of my reaction rate, right? Because first of all, temperature, if the temperature goes up, right? So if the temp rises, there for a second, the reaction rate can go a little bit faster because of it, but then at some point the temperature gets too high, then that's going to break the enzyme, right? If the temperature gets too low, then the reaction rate can slow down, but then it can slow down so much that that's just going to make the enzyme, it's going to make the enzyme not work at all, right? And then pH. pH is another thing. So pH. So if I make the conditions really acidic, so if I put my puppy in acid, he's going to break. If I put my puppy in something alkaline, like a strong base, like bleach, he's going to break. And usually that's because that's going to disrupt, either of these, this is going to disrupt the hydrogen bonds, right? So when I mess with the temperature, when I mess with the pH, really what it comes down to is that I'm going to break these hydrogen bonds. And if I break the hydrogen bonds, that means I don't have a tertiary structure anymore. If I don't have a tertiary structure anymore, then that means I don't have the right shape for my reaction to take place. And if I don't have the right shape for my reaction to take place, that means I'm not going to get a reaction at all. This entire process of breaking these, this is called to denature something. Denature means to break it, right? And generally, if I denature something, it goes all the way back from my tertiary structure down to my primary structure. And we know that since it's just a straight chain of polypeptides, or a polypeptide chain, then he's not going to have the right shape for the reaction to take place. Can I recover from denaturization? Sure, but not all the time. Like some enzymes can refold after a bit, but most of the time they're just gonna be done with it. So then let's talk about a few other examples of types of reactions we're going to see. One is called allosteric. Now, allo means other, okay? And so what that is, it's actually kind of cool. So if this is my enzyme, right, here's my enzyme right here, and then here's my substrate, right? And over here on my enzyme, I erase this little bit, and I don't know what color was there. And there's this little, and then there's, whatever, it's yellow. And then there's this little divot right here. And so this is the substrate that is supposed to react with that enzyme, but look at that. The active site is not the right shape. And so what happens is when you have allosteric, that means you're going to have some type of coenzyme, right? We'll call them a coenzyme. And so now I can take this enzyme, it's kind of deciding. When I take this enzyme and I have this coenzyme is going to fit into another site. When he fits into that other site, that changes the site of my active site, or that changes the shape of my active site. And so now the substrate can fit in. Look at that. And then my reaction can take place. And then whenever the reaction's over, that active site is going to change back into the shape that I don't need it in until I get another coenzyme. And so that's just how allosteric works, is that he changes the shape of my active site so that way that reaction can take place. Then I also have this right here, allosteric, actually has kind of a proper name. This is allosteric inhibitor. There you go. This is an allosteric inhibitor because everything was inhibited until, or this is non-competitive. And then I also have competitive inhibitors. And really all that is that here's my enzyme, right? Now I have my competitive inhibitor right there, and then I also have my substrate. And so sometimes this competitive inhibitor, he's going to go fit inside the active site, which means that substrate isn't going to be able to fit in, and then there's not going to be a reaction taking place. And so that's just some of the things that can happen during enzymatic reactions. But that's a lot of stuff we've covered. So there you go. Enzymes, again, special class of protein. They all end in ACE, or at least a good deal of them. They are always going to work at the tertiary structure. So this is the one I want to live at. That's the definition that you guys have to know, that they speed up the reaction by lowering the activation energy. And of course, how they lower the activation energy is by creating ideal conditions, right? So they create ideal conditions for my reaction. And again, those two reactions can either be hydrolysis or they can be dehydration. Again, depends on what I'm trying to do. And then of course the whole idea of it is that they're specific to a substrate. So that way I only get to do specific reactions whenever I need them. And whenever that substrate fits into my active site, that's called induced fit, so it gives them a little hug. And then these are the things that can denature or that can affect the rate of reaction, temperature being a huge one, pH being a huge one. And then when they actually break that sucker, they denature it. Okay, I hope that helps.